As the Dynamic Biosensors team continues to grow, so does the range of products that we are excited to offer our customers. Our recently launched proFIRE system that you see in the picture before you will be highlighted in our webinar today. Thanks to the extensive work with our SwitchSense DNA-based biosensor technology, we at Dynamic Biosensors have leveraged our experience with one critical element of our experimental workflow to bring you the proFIRE system, and that is the site-selective conjugation of proteins to oligonucleotide sequences. To take a step back, the chemical modification and cross-linking of biomolecules is prevalent in many workflows and is conducted for a wide range of technical objectives, including combining the functionalities of two biomolecules, as you see to the right, such as the formation of antibody-drug conjugates that take advantage of the delivery and uptake of the antibody to targeted cell death induced by the carried drug molecule. A second is the usage of one biomolecule as a structural scaffold, typically DNA origami, owed to its structural resolution and wide design capabilities, to carry a second molecule type. In this figure, we highlight work done with gold nanoparticles. Nevertheless, the concept also applies to the controlled spacing of proteins on DNA origami. And a third, and rather common application, is the attachment of tracer molecules such as fluorophores to enable the fluorescent imaging of targeted biomolecular systems, such as the intracellular actin structures that you see in the bottom right of the slide. As we can gather from the motivations to conjugate biomolecules, the applications for such techniques are very broad and cover the fields of biosensing, including SwitchSense, of course, nanofabrication, and subcellular structural imaging. In these instances, we have chosen to highlight the applications that specifically involve the conjugation of a protein to DNA. For the production of well-defined protein DNA conjugates, we will list some of the main technical considerations to their successful usage. First and foremost is the usage of reaction conditions that maintain functionality of all biomolecular components. Second includes the purity of the reaction yield, as well as the site selectivity, also to maintain functionality as the first point lists. Degree of labeling, yield, scalability, and reproducibility are also all critical parameters for the successful production of protein DNA conjugates. The experimental parameters involved and that affect the outcome of the conjugation reaction, to address the technical considerations that we just listed in the earlier slide, are component feature specific, but also include system parameters. The three main components involved are the DNA, that vary in sequence and length, the protein, whose stability and structure will dictate the linkage chemistry to be used, and the linker used to join both biomolecules. On the scale of the system, the molar ratio of these three reagent components needs to be optimized to control the site selectivity as well as the degree of labeling, meaning the number of DNA sequences that are attached to the same protein. And of course, the buffer system used to run the reaction needs to balance both reaction efficiency as well as enable component stability and functionality. And here is where we come in with the proFIRE system as your optimized workflow to the production of pure protein DNA conjugates. And that is a synchrony of three main elements, as you see before you. The first are the coupling kits to run the conjugation reaction. The second, our proFIRE Purifier device that you see to the right hand of the screen. And of course, the custom-built software for the control of the device, both to monitor the purification run as well as for the analysis of conjugate purity and yield after the completion of the run. Let's start together with the first element, and that is the conjugation reaction itself. Though the type of main components remains the same, so protein, DNA, and linker, the reaction can be customized to combine the modularity in each of these components. And as such, our coupling kits are designed to enable the user to conjugate variable protein types, so using the corresponding conjugation chemistry, as well as variable DNA lengths and sequences. The kits that we provide will contain the DNA with the modification corresponding to the coupling chemistry, as well as the heterobifunctional linker, all of which are pre-allocated for single reaction scale. So for instance, our amine coupling kits will contain DNA modified with an NHS ester, our thiol kits, the malumet modified DNA, in addition to kits with DNA modified for customized click chemistry reactions. For a full listing of our coupling kits, please visit our website and scroll through our reagent lists. Our coupling kits not only come with reagents, but they also come with a step-by-step protocol to guide the user through the conjugation reaction, starting with the activation of the DNA with crosslinker and ending with the incubation of the protein target with the activated DNA. This includes recommendations for duration and temperature of reaction for maximal reaction yields. And as reactions typically do not go to completion, this is where the second element of our workflow comes into play, the purification of the conjugate from the unreacted starting components of the conjugation reaction. In this case, they would be the unconjugated DNA, the remnant cross-linker, as well as unconjugated proteins. Our proFIRE device is an automated liquid chromatography system that enables you to purify your protein DNA conjugates in less than 30 minutes. The device itself is engineered with the objective of optimizing ease of use. The front of the device incorporates a sample injection port, easy to access column attachments, as well as a portable fraction collector. The device is controlled using the tablet element that comes installed with our custom-built software for the real-time monitoring of the conjugation purification run, as well as the data analysis of the chromatogram after the completion of the run. To get started with the purification of your conjugate reaction mixture, first collect your sample using the Hamilton syringe and inject it via the sample injection port. Then to begin the purification run, simply input the DNA length used for your conjugate and click Start. Our easy selection means that customized scripts will run the corresponding purification automatically based on your input. Alternatively, you have the option to modify or generate your own standardized scripts for the purification of your conjugates. And now to describe the hardware features of the proFIRE in further detail. The proFIRE is fitted with HPLC grade tubings and thus handles a wide range of buffer tolerances. The associated chromatography column is an ion exchange column for the analytical scale purification of proteins and DNA. And as you can see in the table to the right, the column can handle a wide range of DNA lengths and protein molecular weights. The user can control the fraction collection volume within a volume range of 200 to 800 microliters, enabling higher resolution fractionation of the elution products. Furthermore, the device enables the user to control the window within the chromatogram over which elution products are collected, yet another feature to enhance the resolution of fractionation. The standardized scripts that come with the device are programmed to select the optimal salt window within the chromatogram that is correlated to the input length of DNA. All information relating to our device specifications can also be found on our website, as well as our product flyer. After the completion of the purification run, you can collect the fractions containing your sample. The data analysis software will guide the user through determining the efficiency of peak separation and thus qualify the conjugate purity, as well as provide an absorbance-based quantification of the conjugate product yield. Here is a snapshot of the output of our data analysis software, whereby the software identifies the individual peaks, protein only, DNA only, and protein DNA conjugate, and recommends the fractions to pool that correspond to the pure conjugate product. The absorbance measurement is conducted at 260 nanometers, is converted into a sample concentration based on the extinction coefficient of the DNA strand per the length of DNA as input in the start screen. Given the concentration and total volume of the recommended fractions to pool, the software will return a conjugate yield in picomoles to allow the user to evaluate reaction yield. And with that, we would have reached the goal of generating pure protein DNA conjugates, which of course opens the doors for us to confidently use our conjugation products for many applications. We would personally recommend that you try out SwitchSense if you haven't already done so. Our biochemistry team continues to develop applications that address the purification of systems beyond the protein DNA conjugates that we covered together today. And these include the purification of protein DNA conjugates, this time using dye-labeled DNA, so bifunctionalized DNA, synthetic molecule DNA conjugates, so for example, attaching fluorophores or contrast agents as well to DNA and the separation of the unconjugated products from the conjugate reaction, the purification of double-stranded from single-stranded DNA, and of course, a growing application that we see with the usage of our device, and that is the purification of DNA origamis that involves the removal of staple strands from the assembled origami structures. To learn more about all these different applications, please stay tuned for our upcoming webinars and events that we are always happy to announce on our social media channels, as well as our website. And with that, I would like to close by thanking our biochemistry team that worked diligently with our hardware and software engineers to bring you the proFIRE system, and that continue to work to enhance the user experience. I would like to thank our sales team that keeps us connected with current and future customers, and to our marketing team, especially my colleague Katarina, that produced all the materials presented to you today. And of course, a warm thank you to you, our global audience, for making the time to attend our webinar today. Please do reach out with any questions or comments to our info@ email address. We certainly look forward to hearing from you. Thank you.